Welcome to Biology Made Easy. What do we have to deal with today? Topic for today is bacteria, prokaryotes. So the objectives for today: describe the structure of a bacterium, identify different types of bacteria, the life cycle of bacteria, and find out if they have any importance. Yes, so bacteria, the most abundant creatures, all right? There are 2,000 million species of bacteria. And prokaryotes, you remember what prokaryotes are? They have prokaryotic cells. The cell has DNA, but doesn't have any recognized nucleus. It doesn't have double membrane bound structures, all right? Okay. And prokaryotes are bacteria and cyanobacteria. And you have 2,000 million species, and they are all microscopic. They are microscopic, but cyanobacteria can occur in large numbers, and so can be seen. As you can see, this blue greenish thing is bacteria, cyanobacteria. In fact, I've seen some in a flower pot. All right, that's cyanobacteria. Well, okay, now let's look at the structure of bacteria. Now, the structure is basically, it has cell membrane, okay? It has a cell membrane, that's a cell membrane, that bounds cytoplasm, all right? And then it has a wall, cell wall. This cell wall is made up of a material called peptidoglycan. And this peptidoglycan is made up of polysaccharides and amino acids. That's the cell wall of the bacterium. And then it has DNA. The DNA is sort of curved into some circular structure. So it's said to have circular DNA. And then it has ribosomes. And so you have the cell membrane, you have a cell wall, and then you have some slimy capsule around the cell wall. This is the basic structures that every bacteria has. All right? But then there are other structures that can be found in bacteria beside the basic ones. And in this, I have made them in red. You see, beside the cell wall of peptidoglycan, it can have an additional wall of lipid and protein. So that's the additional wall, red wall. That's additional wall. And it's made up of lipids, fatty substances, and protein. Then it can have, the cell membrane can have some membrane-like folds inside the cytoplasm. All right? That's an additional structure that's found. Then the other membranes also can be found. Then it has also roundish, tiny DNA called plasmids. You can have one or many. All right? So plasmids are additional structures. Then flagellum, or plenty flagella for movement. Then you have these structures that look like long thread-like structures, hair-like structures. One is called pilus, but plural pili. All right? So these are the extra structures. Flagellum, another wall of lipid and protein. Then you have infoldings, membranes, you have plasmids, pili. that is not found. Some of these structures are found in other bacteria. You can have plenty flagellum, as you can see. All right, these are different types or different structures in bacteria, as you can see. Now, we want to look at the functions of the various structures we have mentioned. So you have the wall, cell wall of peptidoglycan is protective tough. The cell membrane bounds the cytoplasm. The mucilage protects the bacteria from drying out. It also protects it from antibodies. All right, it's protective. The circular DNA determines the characteristics. The cell membranes and infoldings increase the surface area for metabolic reactions. The plasmids makes proteins that confer resistance to antibodies. The flagellum or flagella for movement. And the pili, they are structures that anchor the bacterium and allow genes to be transferred from one bacterium to another during conjugation. We'll look at the function again when we are talking about conjugation very soon. So these are the functions of the various structures that we find. Now we want to quickly look at the classification of bacteria, that is types of bacteria. All right, so the way bacteria reacts to a dye called Gram dye, we have shape. Bacteria is classified by its shape and by feeding method, the way the bacteria feeds. 3 ways to identify the type of bacteria. So now we want to quickly discuss the types. Gram dye. This is a dye that some types of bacteria react positively to it. Look, some bacteria will turn purple. These are the gram-positive bacteria. They turn purple to the gram dye. These are bacteria that have only one wall of peptidoglycan. All right? And these bacteria can easily be dealt with. Example is Staphylococcus, the bacteria that causes pimples and boils. All right? It has only one wall. Then you have gram-negative bacteria. These bacteria, they don't react to the gram dye because they have an additional wall of lipid and protein. They are very difficult to deal with because of the extra wall of lipid and protein, all right? And those bacteria, you have Escherichia coli and then Salmonella typhi. The E. coli causes gut and urinary problems, very difficult. The Salmonella typhi causes typhoid fever. So that is gram dye and bacteria, gram positive or gram negative. Bacteria can also be grouped according to the shapes. You have round-shaped single cells. You have round-shaped that are like bunch of grapes. They are the Staphylococcus. Cocci is plural. All right? Good. You have some bacteria. They are round, but they are arranged like a chain. And you have Streptococcus in here. All right? Then you have this round bacteria that has a capsule around them. The bacteria, there are 2 cells in a capsule, and the capsule is a thick structure that protects the bacteria. This one causes pneumonia. So you have Diplococcus pneumonia. This is how the bacteria looks like. Very, very solid in the capsule. Then you have rod-shaped bacteria. These are the bacteria rod in chains. You have some of them the bacteria has a spore inside it. This one has an oval spore. Very stubborn bacteria, anthrax. That causes anthrax is there. This has a spore sitting on top of the bacterium. All right, this bacteria, Clostridium tetani, this one causes tetanus. Then you have another shape, spiral. All right, look at this bacteria. It looks like a snake. All right, Treponema pallidum causes syphilis. You see, the shape is like a snake. No wonder that it causes trouble. Then you have comma-shaped vibro. All right, and this one too is stubborn one. It causes cholera. Good. So anybody who has cholera has these bacteria swimming in the body. Very bad. Anyway, so we've looked at types of bacteria based on the gram dye, based on shape. Now, the feeding method also separates bacteria. Now, the bacteria based on feeding, you have autotrophic bacteria, saprophytic bacteria, parasitic bacteria. The autotrophic bacteria, autotrophs, they can make their own food. You have two types, photosynthetic bacteria. All right, those who can make their own food, they use sunlight. They have chlorophyll, so they use sunlight and inorganic substances and make food, just like plants do. Plants use sunlight, carbon dioxide plus water, and turn it into sugars. There are some bacteria that do the same. Cyanobacteria do the same, all right? Then you have the chemosynthetic bacteria, all right? These ones are bacteria that will break down chemicals, all right? Chemicals like hydrogen sulfide, release the energy in them, and use the energy to convert simple substances into food, all right? So you have bacterium like Nitrobacter. It's a bacteria that helps in nitrogen fixation. So autotrophic bacteria, they make their own food. Then you have saprophytic bacteria. These ones, they decompose dead organic matter and use the nutrients. So there's bacteria in the soil. There's bacteria growing in the soil. Bacteria can grow in your food or in my food and cause decay and use the food substances causing decay to generate energy, and then also use the nutrients for their benefits. One type of bacteria, Mycococcus, is being named as a saprophytic bacteria. Then you have bacteria that are parasitic. Plenty, we have named them. So look at all the bacteria that causes problems. So now we want to look at the life cycle of bacteria. We should be interested in how they reproduce, all right, so that we can know how to avoid them. We can know how to not let them reproduce. That's one of the reasons why we are studying about these organisms, to know them and to how to use them for our benefit, or how to get rid of them, not to allow them to become plenty to worry us. So the life cycle of bacterium, the bacterium reproduces usually and normally by binary fission. Many of them reproduces by binary fission. A few do conjugation, another type of reproduction, and a few more do budding, another type of reproduction. We will explain each of these. Binary fission, you have one cell. This cell has DNA, and binary fission simply means the DNA replicates. It means it forms another one of itself in the cell, all right? It replicates, and then quickly you have 2 DNAs, if you like, in the cell, and then quickly the cell cytoplasm will divide. The cytoplasm will start dividing. So you have 1 DNA in each. That is binary fission. And bacteria does this very quickly. It divides every 20 minutes. All right, so every 20 minutes, if it has this, and these two also are dividing every 20 minutes, you can imagine. That is the process that we've illustrated here. 1 bacterium, 1 DNA, DNA replicates, cell evangenates, 2 cells are formed. Binary fission, simple. A lot of other organisms also do binary fission. Prokaryotes do a lot of binary fission. All right, amoeba, paramecium, Prokaryotes. Good. Now let's look at the next conjugation, which happens in a few bacteria. Conjugation, simply, it means 2 individuals come together, exchange genetic material, then they sort of move apart, and each one of them starts dividing to form new individuals. Simple. So if this is one individual and this is another individual, they have their genetic material. So they exchange some genetic material. So this one will have some green in here, and this one green here will have some red inside here. So they exchange their genetic material. They start dividing. So this one will produce offspring. This one will have this offspring, and this one so you see, that is conjugation. The two are there, they all have their genetic material, they come together. Conjugation produces individuals that are genetically different from the parents. Meanwhile, binary fission produces individuals that are exactly like the parents, and it's fast. All right? So you can find out the importance of binary fission and the importance of conjugation. Some bacteria also do budding, and a few bacteria. Budding is very simple. It's just that this individual is there and somehow is able to produce part of it, and this is the bud. This part that is producing grows very big, and finally it breaks off. Finally, this is the individual and its bud. Its bud breaks off and grows to become a big thing, individual. This is simply budding. Now there's something that bacteria also do. When the environment becomes unfavorable, the bacterium will form a cyst, will form a tough coat around itself, and then remain dormant. When conditions become favorable, then it will come alive and start reproducing. So when things are hidden in a freezer or a fridge, that the temperatures are cold, the bacteria will be there. But because the conditions are not favorable, they can form cysts. They will remain in. When the conditions become favorable, then they can start reproducing. That is bacteria. Now let's quickly list the importance of bacteria. So you have some assignment to work on, and this is the first one. Suggest the importance of the following: binary fission, cyst formation, conjugation. 2. Could there be any disadvantages in binary fission in bacteria? Explain. This is what we have for today. Interesting bacteria. You know them, how they behave, where they can be found. Everywhere, every environment, there's bacteria. Even in hot springs, there's bacteria. Take care of yourself. Avoid eating cold things that are not neat to avoid salmonella, typhi, and others. So we meet in our next lesson. Goodbye and thank you.